And so I'm going to start by introducing Miss Nicole, who will be Dr. Nicole by the end of the day, I trust. And I would like to point out, in case you haven't really met Nicole, she's the star of today's presentation. You can see her there in this wonderful picture that was taken in Mexico, because Nicole is an IGERT student. And as an IGERT student, her first few weeks at NC State were actually spent in Mexico, learning about the agricultural problems that farmers there face. And as an IGERT student, she really embraced the topic of genetic engineering in society, the case of transgenic pests. And for her cohort, they were studying agricultural pests. And the whole idea of this IGERT was interdisciplinary training. And Nicole embraced this more than any student I have ever seen. And it's really very disparate topics, things like social issues on top of molecular biology, and population genetics and ecology are easier for brains like mine, but those social issues and ethics and policy are really kind of outside of what we normally think about. Miss Nicole, being the go-getter that she is, received a number of awards while at NC State. And I was actually very surprised to see they totaled nearly $250,000. And one of the fun things she did in her spare time, she had so much spare time, that's quite a joke. She was on a debate team with what we refer to as the Thomas Hall Ladies. And they debated the topic of the use of transgenic insects. And not only did they debate the topic, but they won at an international meeting, the International Congress of Entomology. And one of the things I would have to say about Nicole is she's kind of a born leader, but she really wanted to hone these skills. And so she partook in not one, but two of these leadership programs. And these didn't take just a little bit of time. These took major amounts of input in both time and energy. And I think she did incredibly well. The things that I've heard about Nicole from leaders in the field are things like, oh, what do you mean she's not a faculty member? People were shocked to find out that Nicole was merely a graduate student. And some of the things that really gave Nicole the confidence, I think, are things like being a coordinator for BioLunch. She did that for 2 years. She was so popular, she was actually begged to be back for the second year. She was a EGSA coordinator for their seminar series for outreach. And she spent 2 years being a teaching assistant, which also takes an enormous amount of time. But Nicole being Nicole, her research did not suffer from any of that. She has been invited to give numerous lectures, many presentations. She already has 3 publications and 5 more in the pipeline. But I don't want you to think that everything has just been roses for Nicole. Nicole's actually been faced with a number of really significant setbacks, one being COVID. But Nicole being Nicole, the way she handled the lab being shut down, she loaded up her car with the tools of her trade and set up a laboratory at home. So here you can see her newly renovated guest bathroom. And in this case, her guests are beetles. And here's her living room with one of her new coworkers. She didn't want to have to work alone. She's very social. And you can see the stereo microscope where she screened the beetles and gathered much of the data that you're going to be hearing about today. And here's coworker number 2. And because I don't want to eat up much of her time, because she has a lot of really interesting things to talk about, I'm going to hand things over to Miss Nicole. So I'm going to quit sharing and switch over.
All right. Thank you. Let's see. Where did my PowerPoint go? And your screen. Share. Okay, thank you everyone for waking up so early, especially some of my family and others out of state, Marseille and Brenda. It's currently 5:30 a.m. right now in California, and 10:30 p.m. in Guam. So thank you. And thanks for everyone else who also woke up. This is early for me, despite that it's only 8:30 a.m. As all of you probably already know, I'm an interdisciplinary student studying entomology with a minor in genetic engineering and society. And my dissertation, titled Developing, Testing, and Debating Genetically Engineered Insects, covered many topics, including GM insects, gene drive insects, insect resistance to GM crops, and responsible innovation. I'm going to be covering 2 of those topics today, and I'll try to spend around 30 minutes telling you how I developed transgenic Cas9 tribolium beetles, and around 10 minutes on responsible innovation of genetic engineering. So small but mighty, insects are essential to terrestrial life through their ecosystem roles as soil aerators, decomposers, pollinators, and food for other taxa. Humans benefit greatly from these ecosystem services, yet are also negatively affected by their roles as herbivores of crops and vectors of plant and human diseases. Every year, more than 1 billion people are infected by a vector-borne disease, and more than 1 million die. They're also a threat to crop production and competitiveness. This beetle right here alone, the Western corn rootworm, causes over $1 billion in damages every year. Genetic engineering is a powerful tool for us to be able to study these pest insects and pest biology. And in 2012, CRISPR-Cas9 marked a major breakthrough in gene editing. Another useful tool for studying pest biology is the model organism Tribolium castaneum, or the red flower beetle. Red because of its beautiful red shell, and flower because it's a pest of flour or other stored grains. Tribolium is useful as a lab model because it's easy to rear. It's considered a more representative model of pest insects than Drosophila, and it has a good quality assembled genome, and there's already transgenic tools for working with them. Quick recap of CRISPR-Cas9. Cas9 is a protein that cuts DNA, or an endonuclease. And the reason why it's so revolutionary compared to past technologies is that it's guided by this short RNA sequence, here shown in red. And that can be designed to match almost any desired genomic sequence, shown in blue. 5 years ago, I started my first CRISPR experiment. This was a knockout experiment. I took transgenic tribolium. Before you saw the tribolium adults, these are the little larvae that you're looking at. And these ones have green fluorescent protein, or GFP, in their muscles. And I injected these with a CRISPR guide to knock out GFP. And what a gene knockout means is that I've just disrupted that gene enough for the gene to lose its function. And when we first got this to work, we were like super excited. So then I went on to the real experiment that I was super excited about, and we tried to do a gene alteration or insertion. And sadly, that project failed. So why did it fail? Well, here we have the central dogma of molecular biology. DNA makes RNA, makes protein. And when you do CRISPR-Cas9 gene editing, when I inject the embryos with all of my CRISPR reagents, I can provide Cas9 as any of these forms. I can inject DNA that codes for Cas9, I can inject RNA, or I can inject Cas9 protein. Well, unfortunately, back then, there weren't any reliable sources yet of Cas9 protein. Nowadays, you can get it no problem. It's a little bit expensive, but it's totally reliable. Back then, we didn't have that option, so our next best option was to make transgenic tribolium. Therefore, those insects should already have the Cas9 protein ready to go in their eggs. However, they're only going to have it in the eggs provided that we use a gene promoter that makes sure it's active at that time. And for example, so here we go. This is a beautiful picture I found of Tribolium embryogenesis. The embryo's forming, and slides A and B are the important ones here. That's the formation of what's called the blastoderm. When I'm doing gene editing, it's essential that I inject these eggs with all my CRISPR goodies at the right time. The sweet spot for Tribolium is 2 to 4 hours old, and that's when the eggs haven't undergone cellularization yet. And so again, in order to increase the Cas9 cutting, we want to have protein in the egg at that sweet spot between 2 and 4 hours old. And so I use the word promoter. For those of you that don't know, a very basic level of a gene is that you have a promoter. It's kind of like an on-off switch for the gene. It tells the gene when and where to be expressed. And then you would have the coding sequence, which is the part that's actually going to turn into the protein. And then you have a terminator, which is the end of the coding sequence. For example, you could have an eye-specific promoter. So that gene is only going to be turned on in an eye. Or you can have a full body promoter. Well, for us to drive our Cas9 transgene, we decided to go with a constitutive full body promoter that we know should also be on in very early embryos during that sweet spot at 2 to 4 hours old. We decided to go with polyubiquitin and alpha-tubulin. And this was great because these promoters have been used already to drive transgenes. That's the photos that you're looking at. So I didn't have to do any testing with these promoters. I already know they're gonna work to drive a transgene. And an added bonus was that we know polyubiquitin and alpha-tubulin transcripts get maternally preloaded. What that means is insects do this cool thing where the mothers provision their unborn embryos with transcripts of important genes. So that's just an added bonus for us because not only is the gene going to be turned on in the embryo very early, but the mothers are going to provision more of the Cas9 transcript into the embryos. So that's great for us. And how did I get this Cas9 gene into the Tribolium genome? Because I couldn't use CRISPR, right? So instead, I used the piggyback transposase system. How this system works is it's a cut and paste system. The transposase, in this case, is the protein that's doing the DNA cutting. And where it cuts is at these piggyback recognition arms. There's 2 of them here, the arrows. And within those arms, within the sandwich, I can put whatever genetic cargo I want. In this case, it's going to be my Cas9 gene. So the cargo and the recognition arms get chopped out and then they get pasted into the genome. And where it inserts is semi-random. Piggyback recognizes a target sequence TTAA, but that sequence is in a lot of places in the genome. So there's lots of places it can land, and we don't exactly have control over it, but that's okay with us. So again, my genetic cargo, I'm actually gonna have 2 pieces. I'm gonna have my Cas9 gene with my 2 promoters, either, I'm gonna have 2 different constructs with either the polyubiquitin Cas9 or the alpha-tubulin Cas9. And that's gonna make the beetles so that they have Cas9 all over in their bodies. However, I can't visualize that at all. Even though they're gonna have Cas9 everywhere, I can't see that. So a tool that we use in molecular biology is what's called a marker gene. And here I've decided to use the green fluorescent protein, or GFP. It originally comes from a jellyfish. And for the marker gene, I'm using an eye-specific promoter. So they're gonna have green glowing eyes. So when I use this marker gene, after I do my genetic insertion of the Cas9 gene, then I'm gonna be able to know that I succeeded when I see green glowing eyes. And what tribolium strain did I use for this? I used the piggyback transposase helper line, M26. It's called a helper line because it's transgenic and it already has the piggyback transposase in it. In this case, the transposase gene is on the X chromosome and it's marked by DS red. So they're gonna have red fluorescent eyes. And based on the size of my cargo, we expected to see around a 14% transformation efficiency. So out of all the embryos that I inject and that survive, we expect around 14% of them to have green eyes, which indicates the Cas9 transgene. To give it a little extra oomph, I also injected some helper plasmid, and so actually our transformation efficiency could possibly be a little bit higher. How this works is I let the beetles lay eggs for 2 hours. I collect those eggs. Here's a size reference. This middle photo, this is my egg basket. That's probably around 300 tribolium eggs in there. This is a Petri dish, little plastic Petri dish. And then I line them up on a glass slide and I micro inject them with a glass pulled needle right here. And I can use red dye to help me visualize that, because in actuality, these are really small, even when I'm viewing them under the microscope. Unfortunately, with that green eye marker, I can't visualize that in the beetles that I inject. I can only see it in the next generation. So the first thing I did was I let them go through their life cycle. They become a larva and then a pupa, which we'll see later. And an adult, that takes around 32 days. And I gave them a mate, and then I put them in vials and put them in the incubator for about around a month. When I mated them, I outcrossed them to vermilion white. It's a white-eyed mutant strain. It's the parental line of M26. And the reason why I didn't just mate them to more M26 is now that I've done my injections, I don't need that piggyback helper anymore. I want to start getting rid of it. And the useful thing about M26 is, like I said, the transposase is on the X chromosome. So if I keep selecting males to do my crosses each time, I can pretty quickly get rid of that transposase gene. And I had success, yay! We like success. I had my 2 different injection constructs. I had my eye promoter, or eye marker, and then I had my polyubiquitin Cas9 or alpha tubulin. This column is the number of adults that I got out. These were all the injectees that survived. This was the number of fertile crosses. So not all crosses were fertile. And then this was the number of offspring that I ended up screening. So you can tell that took me a little bit of time. And this is the number of successful crosses. And from this number of successes, I can calculate the transformation rate, which you can see is between 2 and 3%. And remember, we were expecting around a 14% success rate. So at first we were kind of concerned or perplexed by this, but we came up with an idea of why this is, and we'll go over that a little bit later. Most of my screening was done in the pupal life stage, but I thought these pictures of adults were just really cool. I like how their eyes look. So this is an adult beetle. Here's its little antennae here. Here's the eye. And this is under white light. And then on my fluorescent microscope, I can use a DS red filter and view red fluorescence. So here we go. And then this is what success looks like. Got my green glowing eyes. Hooray! And so remember I showed you that we had 5 successful crosses for polyubiquitin. Cas9 and 2 for alpha tubulin. Well, out of these crosses, I only got a small number of offspring that actually had the green eyes. So one of the crosses gave me 1 individual, another one, 3, 3, 4. So in all, I only got 20 individuals that had those glowing green eyes. But remember, I used the piggyback transposase system, so we don't know where that Cas9 transgene landed in the genome. Each of these that came from the same individual, so if I got 3 offspring from a single injectee parent, it's possible that those 3 individuals from the same parent might have the same insertion area in the genome. The transgene might be in the same spot, but they could also be different. And at this point, we don't really know. In order to figure that out, if they're all unique, then we would have to do sequencing. And so in the meantime, before I was going to do sequencing, I wanted to keep all of them separate. So I started with 20 individuals. I started a different line, a unique line from each of those possibly unique individuals. And I first kept outcrossing to the vermilion white mutants. That way I could get rid of the piggyback transposase. During that process, I also started discovering that I had multiple insertions of my transgene, of my Cas9, just for a couple of the lines. So I had to do extra outcrosses for those. But once I was sure that I got rid of the piggyback transgene, and I also only had a single Cas9 transgene, then I started brother-sister mating them for multiple generations until I knew that I had homozygous lines. This whole process took me over a year, and during that time, I lost 10 of the 20 lines. And why we think that is, well, first of all, genetically modified organisms usually aren't as fit as non-modified ones, but also it's probable that my transgene landed in an area in the genome where it disrupted a really important gene and it ended up being lethal to those beetles. I also became interested in fitness because some of them were not laying that many eggs. I decided to measure direct fitness, which is the mean number of offspring that a single female can create. It's not as good of an assay as competitive fitness. However, the number of eggs that they were laying was so low for a couple of the lines that that assay would have been impossible for me to do. So I decided to go with direct fitness, and I'm going to take you across the x-axis. I ended up with 10 tribolium lines, or Cas9 lines, numbers 1 through 10, but then the 10th one, right after the fitness assay, before I could do any other experiments with them, they ended up dying. So here I called them D for dead. And then I had 3 control lines, Georgia 2, which is a quote wild type, but also very inbred line, and VW, which is a white eye mutant strain, which is the parental strain of my Cas9 lines. And then A19, which is those green glowy guys that you saw at the beginning with the GFP muscles. Another thing I should tell you about this graph is numbers 1 through 4 are the polyubiquitin Cas9, and then 5 through 9 are alpha tubulin. So just looking at it, you can see the alpha tubulin looks significantly to have significantly higher fitness than some of the polyubiquitin, but not in all cases. With the error bars, some of them do overlap. And then not surprisingly, the one that ended up dying looks like it has the lowest. The cool thing about this though, is I was very excited to see that some of my transgenics were neck and neck with some of these controls. VW's doing a lot better than everybody else, but these Georgia 2 and A19, they're right around the same. So that was great for me because I was a little bit concerned that their fitness would be lower. And at the very end of this, I'll tell you which of my favorite lines are. Like I'm gonna pick 2 favorite lines, so fitness will come back up in a little bit. Now that we tested the fitness, I wanted to find out where those transgenes landed in the genome. The protocol I used for this was called inverse PCR. How that works is you take the genomic DNA, you chop it up real small, then you ligate it into small circles like this, and then that allows us to be able to sequence all the way around that plasmid. What you expect to see when you get your sequencing results is a little bit of the piggyback recognition arm, that TTAA site, and then directly after that should be Tribolium genomic sequence. However, that's not what we got. Instead, we saw random bits of my plasmid right after the TTAA site, like a little bit of the plasmid backbone, or a little bit of EGFP, a little bit of Cas9 gene, and all of them were different between the lines too. And so we had to come up with an idea of what the heck happened. And remember, this is a cut and paste system. We think what happened was this. We think that, or some variation of this, the transposase cut my cargo with the recognition arms, and possibly I injected way too much, or like too high of a concentration of my donor plasmid, and therefore before the cargo could insert into the genome, it first inserted back into another plasmid, and you got this weird concatemer thing where I've got, you know, a full transgene in there in the middle, but some sort of weird gobbledygook on the outside before then inserting into the genome. But they were also all different, so they probably are all different variations of this. So that made things a little bit tough, but I'm just glad at least it worked, and they have Cas9. What we turned to instead of inverse PCR was we sequenced their genomes actually. And thus far, we've been able to identify the location of 5 of them. 1 is on LG7, 3 are on different areas of LG9, and then another one aligned to an unassigned scaffold, which is still informative because we know it's unique from those other lines. And what's cool is that, so we've identified 5 of the 9, and each of the ones, even if they were siblings, if they were offspring of the same G0 injectee, remember I showed you that table and I said, well, some of the siblings might be the same insertion location, but we're not sure quite yet. We're gonna have to figure that out. Well, this shows us that they actually are different. So far, all of the lines are all unique from each other. So I'm glad that at the very beginning, I kept them all separate. Now that we know the location and we know their fitness, I wanted to see, well, how well do these Cas9 lines work? I mean, that's the real purpose of this, right? Is to increase Cas9 cutting efficiency. To do this, I did an EGFP knockout experiment. I crossed my Cas9 lines with the green eyes out to our A19 line, which has green muscle and also green eyes. So the offspring from that mating is gonna be heterozygous for the EGFP muscle, and they're gonna be heterozygous for 2 copies though of EGFP eyes. As a control, I also crossed my Cas9 to the VW wide-eyed strain. And this is what knockouts look like. What you're looking at is the dorsal view of a pupae. So the eyes are gonna be on the other side, and this is their back. I had non-knockouts where there wasn't any muscle fluorescence missing. There was some that had low levels of knockout where you could just see small patches here that are missing GFP expression. And then I called medium knockouts were ones that where they were at least 50% knocked out, and then high knockouts where it was at least 95% knocked out. And here's my data, and I was very excited by this. The first column is the promoters, so either polyubiquitin labeled in gray or alpha-tubulin in white, the Cas9 line numbers 1 through 9, and I've ranked this entire table from a high level of knockout to a low level of knockout. The percent of injectees that had a knockout. And for this, it was no matter if they were low, medium, or high knocked out, that's what this one is. And look, 87%, I was very excited by that. However, some of them, alpha tubulin number 5, not doing so hot, 27%. I was also interested to see, well, what about if we just consider the ones that were medium and high levels of knockout? The reason why this is important for us is because when we're doing gene editing, usually we're not interested in just knocking out in, or gene editing in the injectees. We really want that gene edit to be heritable. We want it to be in the offspring. And so if we have medium and high levels of knockout, it's more likely that that gene edit we made was heritable. So that's why we were interested, and I've done this table the same way. You can see the numbers are a little bit lower, but the top 3 performing lines, 4, 2, and 6, 4, 6, and 2, they're pretty much the same. And also we kind of see a trend where polyubiquitin's performing a little bit better than alpha tubulin, but thus far, I think 4, 2, and 6 are my favorite lines. I also looked at EGFP-I knockout. Unfortunately, when I crossed together my Cas9 lines and the A19 lines, even though those are both wide-eyed mutants, they're different wide-eyed mutants. So when I mate those 2 lines together, they do end up having wild-type black eyes, and it makes it hard to see the GFP a little bit. So I did not rank the eye knockouts the same way I did with the muscles. I didn't say low, medium, high. I just said, are they knocked out or are they not knocked out? And also their eyes are really tiny, so it's hard anyway to view knockouts. This is a picture of just what the knockouts look like. I thought this one, he just looked cool. He had one eye where it was like completely knocked out. Most knockouts, it was like small sections or like half an eye. And again, this table, I have ranked it from high to low knockout. And here you see a complete distinction between the polyubiquitin Cas9 lines and alpha tubulin. And now I want to bring back fitness again, because I don't want to have to keep, and I'm sure Marseille doesn't want to have to keep 9 of these lines forever. So if I had to pick just 2 lines to keep forever and ever, which ones do I keep? So here's all of my data together. These top 2 are the first tables you saw. This bottom left table is the eye knockout. And then here I've brought back fitness. And this was where it gets a little interesting again, because for the gene editing, polyubiquitin looks a lot better. But for the fitness, polyubiquitin, not so good. However, I still think that lines 4, 2, and 6 are my favorite. But especially because of the fitness, I should definitely keep that line 6. Even though it didn't perform as well with the gene editing, like we see 4, 2, and 6 is 3rd good, 6 is 2nd good, 6 is all the like halfway down with the eye knockouts. However, line 6 did pretty good with the fitness. So all in all, I think I'm going to keep lines 4 and 6. And so also, if anybody, after I publish, if anyone requests these lines of me, I would probably ask them a little bit about their project. And that way I could tell them if they should take line 4 or 6. because if they're doing a gene edit where fitness is a big deal, then I think it's totally okay to sacrifice a little bit of gene editing efficiency in order to have that higher fitness. But if they're doing a project where fitness isn't as big of a deal, or if they're going to be outcrossing a bunch like right away to get rid of the Cas9 transgene, then it's totally probably fine that they have the line 4 where it has a little bit lower fitness, but then look at that 87% efficiency. Also, what I didn't already tell you is that the pilot study of this, I was using another guide that was like, we knew to be medium efficiency, like around 50 to 60% efficient. And I couldn't even see a difference between the lines. They were all up around 100% efficiency. So in order to do this experiment and be able to see differences in between the lines, I had to purposefully choose a guide that I knew was not as good. Previously, it had been around like 30% efficient. So that's very exciting. But remember, why we're really doing this is we're building tools in this model organism because we want to be able to transfer some of those transgenic tools over to even bigger, badder pests, like the billion-dollar beetle Western corn rootworm. And that brings me to the second part of my talk. When we use transgenics to combat big pests like the billion-dollar beetle, it's called genetic pest management. And as a GES student, or genetic engineering and society, I think it's really important to not lose sight of the bigger societal impacts that these technologies are going to have. So this chapter of mine was called Responsible Innovation in Genetic Engineering Research. What does it mean to early career researchers? Now, as long as genetic modification has been a thing, so too has been public controversy around the technology. Back in the 1970s, most of the technology was in, or applications were in GM crops, and this sparked what's called the GMO debate. And this was just the cutest poster that I've ever seen about the GMO debate. Here we have the cute, innocent corn with the googly eyes and the butterflies and flowers. And then here we have the genetically modified Frankenstein corn, Frankenfoods, and the comets raining down to destroy the earth. So, but all jokes aside, this is really important. And back in the 70s, developers were so concerned about answering all the biosafety questions that they kind of put to the side some of the ethical and societal questions. But now that the technology is advanced, we're bringing those questions back to the forefront, especially in the GM insect world and also gene drives. People are really thinking about those bigger societal questions. And even though a developer like me might think that my little beetle babies are very cute, I can certainly see where people are coming from when they think that they're scary and dangerous. For example, in 2009, the Florida Keys Mosquito Control District started thinking about the possibility of using genetically modified mosquitoes in order to control the mosquito population and protect people against dengue and Zika virus. In 2012, they made the decision that they were gonna pursue the technology. So they started informing the public. And the way they did that was through radio talk shows, through the local newspaper, and public events. They also did a couple surveys to see what the community thought about the technology. And for the most part, it was pretty positive. They found around 20% of the community were against GM mosquito releases. And they had gotten approval by the FDA, and they were going to start a trial of these GM mosquitoes. However, it ended up getting canceled because there were so many protests around the technology. And the former executive director ended up he had 3 takeaways from his experience in this situation. He said that 1, support from a numerical majority doesn't equal public perception of a majority. 2, the public should have been provided data and answers earlier in the process. 3, they should have met with individual residents before the opposition group did. And this, I think, is where responsible innovation could have helped mitigate some of these challenges. Responsible innovation encourages upstream public engagement, and it also encourages developers to really think about the assumptions they have around the technology and think about the wider impacts. So what the heck is responsible innovation? There's multiple frameworks for this, but the most cited one is this. It has 4 components: anticipation, inclusion, reflexivity, and responsiveness. And anticipation means considering the possible desired futures of the technology, as well as considering the intended and unintended impacts. And you're supposed to do that before the technology is even fully developed. Reflexivity is thinking about the assumptions that you have and your values, worldviews, motivations that you're bringing to the project. Inclusion is advancing deliberation among publics, stakeholders, and regulators to bring different values and norms into the decision-making process. And responsiveness is responding to those other 3 components, changing the trajectory of the technology as that information comes in. Responsible innovation has been applied to genetic engineering or GM products by many scholars, including GM crops, GM livestock, and there's even been 2 papers applying it to GM insects. There was a mosquito paper and a Drosophila suzuki paper. The framework's transferable across sectors. So in Europe, they actually use it in policy and funding of biotechnologies. However, in the US, it's mostly just stayed in academia. Well, in the last 5 years, there's actually been a lot of papers about responsible innovation for genetic engineering, looking at the perception that's held by stakeholders and established researchers. However, early career researchers have been largely overlooked. And I think this is important, especially for gene drive insects, because gene drive is mostly being researched in academia, and therefore the people who are directly working on this technology is graduate students, early career researchers. So I think it's important to have their take on this. So I set out to do that. I did semi-structured interviews. I interviewed 10 graduate students. They were all one-on-one interviews and each 1 hour long. I had 12 open-ended questions, which were asking them what they thought like each of those components meant, what were the roles of scientists and the public in decision-making processes, and so forth. This project was part of a larger grant funded by the NSF called Cultivating Cultures of Ethics in STEM. And the PI on that project was Jennifer Kuzma. And there was a couple co-PIs, and it was all people in our GES center. How this works is I do the interview, which is recorded, audio recorded, and then it's transcribed, and I import it into a program called In Vivo that lets me code the data. What coding means is I search through the transcripts for different themes using a code book. And my code book, which has all my themes in it, was built based off of the responsible innovation literature. And also when I'm doing the coding, what I find is usually like a 1 to 2 sentence area where they're talking about a theme. I ended up with around 30 themes, but I've simplified it for this talk. These were some of the most referenced themes. And what was interesting is, in addition to talking about what responsible innovation, what those 4 different components looked like to students, they mostly talked about the challenges and opportunities of doing responsible innovation. They were mostly focused on the feasibility of incorporating it into their graduate research. And I've picked out 4 of the most salient ones for me: onus, or the duty or responsibility; skills or knowledge; authority; and collaboration. Here I have the number of students out of 10 that mentioned that theme. Here's the number of references, so the number of times they talked about that theme. And then I split the number of references if they were talking about it either as an opportunity or as a challenge. And you can see they're different. So students viewed the onus as the biggest opportunity for doing responsible innovation. They thought that their being on the ground and having their actual hands on the project and the focus on the project made them more capable, even more so, they said, than professors at doing responsible innovation, because professors have to split their time and their focus between so many different projects in their labs. And so they might not see what needs to be seen, they said. And however, skills or knowledge they found as a challenge. So despite the fact that they felt this duty and responsibility to be stewards of the technology, they said that their ability to do so was hampered by their lack of skills or knowledge in social sciences or doing public engagement or things of that nature. And so they called for more training in ethics and also social science methods, and especially skills in communication. They said that was important for 2 fronts. If they were gonna collaborate with, like, have an interdisciplinary team, they said they were not confident with communicating with people from other disciplines. And then even more so, on the other front, they said that they don't feel confident in doing public engagement, also because they couldn't communicate their science. So they called for more training in those areas. Authority was another challenge that they listed, and they said it was because the rigidity of the dissertation process hampers their ability to do the responsiveness aspect. So remember, that's the component where you're responding to that input that you got and changing the technology direction or changing the research a bit to better fit and align with what society wants. And they said in a dissertation, you don't have the time or the freedom to really be able to do that. Once you're locked into a project, you're locked in. We can't do that as graduate students. However, collaboration, which is the last one here, was seen as an opportunity to be able to facilitate them in situations where they had issues with authority or the lack of skills or knowledge. They said collaboration with people from other disciplines would help fill in the gaps of their skills or knowledge. And also in areas where they might not have the funding or their major professor is just not interested at all in doing projects like responsible innovation, collaboration can help give them authority to do responsible innovation. So their overall takeaway from the interviews was that they saw a need and potential for responsible innovation, but as it is right now, they think it's too forward thinking. So they really called for some of those major challenges to be addressed. However, they still saw it in a positive light and outlined some of the opportunities. And this is where I feel very fortunate, because as an IGERT student and a GES fellow, I've felt much less of that burden of a lack of authority for asking these kinds of questions. I feel very comfortable in the GES Center asking these questions, and I'm encouraged to do so. One of my chapters was this social science chapter, and I have an additional paper that I'm also going to be publishing from that work. And I would not have had that authority or the skills or knowledge if I didn't have this interdisciplinary training. So I'm just forever grateful for the program. And that brings me to my acknowledgement slide. Thank you to my committee, Marseille, Jennifer, Max, and Fred. Also, thank you to the senior people in my lab that helped me with all of my experiments, especially early on, Nathaniel, Clay, and Will. Thank you to the undergrads who helped me collect data, Ashley, Lyric, and Lauren. And thank you to the Thomas Hall ladies for making graduate school so much fun. And with that, I will take any questions.
So we do have a question already from Jennifer. Let's see, she says, Nicole is now a social scientist too, after her very nice chapter on responsible innovation. So that's pretty cool. And Jen asked about, she thinks that it's possible that she's confused about the meaning of onus. She said that she thought it was interesting that students didn't identify onus as a challenge, or that she wanted to know, why is the idea of duty not also a challenge?
Like as if they felt burdened by it?
Yeah, I'm curious because, you know, for example, I feel an onus to recycle, yet there are challenges in actually recycling that I could identify very easily. And so I found it curious that the students weren't identifying the challenges associated with the duty that they felt.
So I talk about a little bit in my written dissertation some of the caveats of my study, and I think that might play into this a little bit. These students, before I interviewed them, they went through a 3-day training process to be student moderators for a responsible innovation focus groups. And I talk about that we might have biased them a little bit, as like maybe we had some sort of call to arms, where they're especially viewing this in a positive light and like, oh yeah, that is a good idea. We should get on this. You're totally right. Let's do it. This is great. We believe in it. So it might have just been a response of they hadn't like gotten that far yet to really think about how the onus could be a burden. Although I think it kind of is addressed, because again, they kept saying they viewed it in a positive light, but they also pointed to all of those challenges. So yeah, basically it's complicated, but you're right, that is the caveat of the study.
So there's a question in the chat from Jason Delborn, and he wants to know, how does responsible innovation change the way you think about your own technical work with CRISPR and the beetles?
So I have thought about this after I finished that project. I'm like, oh no, now I kind of feel like a hypocrite, because a big part of responsible innovation is that you're doing this before the technology is even fully made, right? And also, I never did public engagement or interfaced with the public at all. So even though I studied the topic of responsible innovation, did I really do it? And I think that's where Adam Kokotovich gave me a little more, made me feel better about my situation when he gave a colloquium a couple weeks ago, because I asked him, well, what about graduate students who feel like it's hard for them to do public engagement. And he said, you know what? Well, that's just going to make people stop in their tracks and feel like they can't even move forward or towards responsible innovation at all. So at least if you try a bit and gain more knowledge on it and do any little bit of public engagement, then you're moving in the right direction, essentially. And so I might not have exactly done responsible innovation during my dissertation, but now I feel prepared moving forward wherever I end up getting a job later to keep that in mind. And it's definitely a goal for me to move towards.
So I think we have time for one more question. Nora asks about looking back over your graduate trajectory. What changes would you like to see in graduate education so students feel greater authority to enact something along the lines of responsible innovation?
Yeah, so 2 parts. I think funding. In the NSF, when you write grants, you have a broader impact section, but even that section is just very narrowly focused on the science impacts. If some of the funding agencies could include responsible innovation topics, I think that would give students motivation. Also at a university level, I think community is important. Like I said, I feel very fortunate to be a part of the GES Center. If centers like that, interdisciplinary centers, were available to students, it gives you the space and the drive to get these new skills and do projects on them. And it gives you access to collaborators who are enthusiastic about that topic. So I think interdisciplinary centers and funding.
Excellent. Well, since we are almost at the end here, and we have to give Nicole a little bit of time to prepare for the next portion of her grueling day, let's all thank Nicole with our, let's see what we call it, a virtual clapping. Thanks so much. I really enjoyed your talk and your cute little beetles, but I might be slightly biased.
Probably so.
Thank you, everyone, and thanks, Nicole. And we will resume on another channel in about what, maybe 10 minutes? Does that work for everyone?
Works
I should say the committee members.
Thank you, everybody, for coming. I appreciate it.
See you in a little bit.
Hey.
Thank you. All right, before I log off, I'm gonna try to save this chat.
Yeah, I was gonna try to do that too. But you probably have more control.
I am technologically illiterate.
Okay, I did a little save.
Let's see.
I just don't know where a save goes.
Chat saved. All right, I think there's a Zoom folder.
And I'm going to try to send it to you in an email.
Okay, sounds good. All right, so I'm gonna close this out and go potty and be back.
Okay, great job.
Thank you. Bye.
See you in a few minutes.